Well, thank you for the honor of addressing the Academy this morning. In the next half hour or so, I'll tell you about recent work to harness the immune system to treat and hopefully cure cancer. Start with some disclosures. So, and you can see my spouse was the main culprit on that slide. So, you've probably all heard that cancer is 200 different diseases, and now you've seen from the first two speakers that cancer is many hundreds of different diseases based on its genetic complexity. This graph is taken from a publication from Harvard and the Broad Institute looking at a variety of human cancers and their somatic mutation rates. You can see that some of the carcinogen-induced cancers, such as melanoma and lung cancer, are on the far right-hand side of this graph. With the highest mutation rates. This mutational heterogeneity in cancer does create challenges for oncologists, and you've heard how, as a physician, you can block one of these signaling pathways, but rapidly others emerge to take over and drive the cancer cell. However, from the standpoint of an immunologist, these mutations create opportunities for immune recognition, and in a sense, they provide a common denominator for cancer immunotherapy. Now, the adaptive immune system in many ways is an ideal anti-cancer agent. It has diversity, which is almost limitless. And so through recombination events, T cells and B cells through antibodies can recognize many different antigens. The immune system is also highly specific. It can distinguish minute chemical alterations. So a chemical modification as small as a single methyl group or a single phosphate group can be a go, no-go decision for immune recognition. And finally, the immune system has memory, and this property makes it, I think, different from any other cancer therapy that you can imagine, because the immune system is a living entity, and it should be able to evolve as the genetic complexity of the cancer evolves. It should be able to keep pace with changes in cancer. If we can properly train the immune system to recognize cancer, that education and that memory should be able to last a lifetime. So why then do cancers form? Why do they metastasize? Well, we've learned over the past 10 years or so that actually the balance between the immune system and cancer is one of tolerance rather than rejection. So the immune system should see cancer as foreign because of all of these somatic mutations, but it doesn't. It sees it as self. How does this happen? How does this occur? We think now of cancer as an immune disorder. It's not a global immune disorder because most of our patients are immunocompetent, but this disorder occurs on a local level within the tumor. So within the tumor environment, and Jeff showed you an image of this, there are a lot of things going on. There are regulatory immune cells such as Tregs and myeloid-derived suppressor cells. There are immunosuppressive soluble factors called cytokines, such as IL-6, IL-10, TGF-beta. And on T cells, the major immune cell that fights cancer, there are inhibitory receptors that we term checkpoints. Now, what are the potential clinical solutions? How can we get around these various barriers? Well, in terms of these regulatory cells, we might be able to devise potent vaccines against cancer. And that's something that I'm not going to have time to talk about today. But another solution here is to take the T cells out of the suppressive environment and manipulate them in the laboratory, to engineer them to be cancer killers, to grow them up and then transfuse them back into patients. This is called adoptive T cell transfer, and I'll touch on this briefly in the talk. But what I'm going to spend most time talking about is the solution that we term blocking immune checkpoints, to use antibodies to basically block these inhibitory receptors that would otherwise suppress anti-tumor immunity. So adoptive T-cell transfer has been most studied in melanoma. Melanoma, for reasons that we still don't quite understand, is the most immunogenic of all human tumors. It provokes very strong immune responses, and so it has been the case study for development of most immunotherapies. Adoptive T-cell transfer started many years ago, and you can see that these publications go all the way back to the 1980s, and at that time we were taking solid tumors, bringing them into the lab, dissociating them with enzymes, selectively growing out T cells, and then transferring them back into the patients. And we did see some fairly dramatic responses, but this is really the hallmark of personalized therapy. You can imagine the amount of effort that went into treating each one of these patients. Then we tried to refine this treatment, and in refining it, we anticipated we would do better, but in fact, we did worse in terms of the clinical response rates. And so when we got to the point in the lab where we had all the technologies where we could grow individual T-cell clones, each T-cell just recognizing one tumor antigen, and we gave those to patients, the response rates dropped dramatically. This occurred because tumors can evolve to escape immune recognition, and this is a perfect example from a study by Cassian Yi and colleagues published in PNAS in 2002, where they used T-cell clones that were grown from the blood of a melanoma patient. They knew that these T-cells recognized the antigen MART1, which is a melanoma-specific protein. They transferred those T-cells back into the patient and saw tumor regression. But then they saw tumor recurrence, and when they biopsied the recurrent tumor, they found that the target antigen was no longer being expressed. And this was very specific because when they looked at other melanoma antigens in that patient, their expression hadn't changed. So on the one hand, good news, okay, immunotherapy had an effect. It killed the cells that were expressing the target antigen, but then the bad news is that there were many other tumor cells there that did not need to express this non-essential enzyme, and they grew out. Now, this entire area of adoptive cell transfer has evolved recently to what some people consider to be an off-the-shelf reagent, chimeric antigen receptors, or CARs. The concept for CARs was originally published in 1989 in PNAS by Zelig Eshar. He showed in preclinical models that you could take the extracellular domain of an antibody, which has very high specificity, and you could merge it with the signaling domain of a T-cell receptor. And so in essence, you had a product which, on the one hand, had properties of immunoglobulins, on the other hand, had the function of T-cells. It should be the best of both worlds. Now, it took all this time since 1989 for the technology, for the science, to catch up with the ideas. But now, recently, we've seen publications from Carl June's groups and others using these chimeric antigen receptors directed against the molecule CD19, which is expressed on certain kinds of blood cancers, leukemias and lymphomas, to actually cure patients with very advanced disease. And this is showing you one example from one of Carl June's New England Journal papers, showing a patient with B-cell lymphoma, and you can see on this CT scan, this patient has many enlarged lymph nodes under the arms, and they are disappearing within the first few months of therapy. Bad news, however, is that even with this technology, antigen-negative variants could grow out. And so Carl has shown that when some patients relapse, they have lost expression of the target antigen. And so this brings us around now to a discussion of immune checkpoint blockade. So this diagram depicts what we call the immune synapse. It's where the T cell encounters antigen-presenting cells, such as monocytes or dendritic cells or tumor cells. And it's a complicated scenario, but it can be boiled down essentially to two signals. So T cells function according to two signals. The first signal, signal 1, is the specific recognition event from a unique T cell receptor binding with an MHC molecule, a tissue typing molecule essentially, and a peptide derived from the tumor cell. And Bert showed you a diagram of this earlier. So this is recognition, but then there needs to be a second signal to tell the T cell what to do. It can either become activated or suppressed. And so that's where all of these co-regulatory pathways come in. The first of these co-regulatory pathways to go into the clinic was the inhibitory receptor CTLA-4, which binds to CD80 and CD86. This move into the clinic was based on preclinical work in animal models by Jim Allison, showing that by blocking CTLA-4, you could cause the regression of advanced murine tumors. And in 2011, a drug blocking CTLA-4, ipilimumab, an antibody, was approved for use in patients with advanced metastatic melanoma. Now, this was great news for melanoma, which is my clinical specialty, because it was the first time ever that a drug had been shown to improve the survival of patients with advanced metastatic disease in a randomized trial. And here I'm showing you the pivotal graph from that trial, in which you can see that patients receiving anti-CTLA-4 had much improved survival compared to the control group that was receiving a peptide vaccine. Also notable is the tail on this curve, which is plateauing at about 20% overall survival. These results from the 2010 New England Journal paper by Hody and colleagues have recently been updated. Many of these patients are still alive 10 years later, and so it appears that that tail on the curve maintains itself for a very long time. Now, most of these patients on the tail were not cured of their melanoma. So they still had visible tumors on scans. They continued to go to their oncologist and be scanned every few months. This suggests that we had reset the balance between the immune system and cancer in a really very enduring way. Well, why not just stop there? CTLA-4 was extremely important and continues to be important as the prototype drug in this class of checkpoint blockers. However, as you might imagine, by blocking an inhibitor on immune interactions, we saw side effects with inflammation in normal tissues, autoimmunity per se. And so with this particular drug, the rate of serious immune-related side effects was about equal to the rate of prolonged overall survival. And so we knew that we needed to keep looking for other agents in this class that might have an improved benefit rate and a reduced toxicity rate. And so we decided to look at the PD-1 pathway as a new strategy for checkpoint blockade. We were interested in this pathway because Li-Ping Chen had shown in 2002 that PD-L1 is selectively upregulated in many human cancers. And along with work from other laboratories such as Gordon Freeman and Arlene Sharp at Harvard, there was evidence that by blocking PD-L1, you could cause the regression of established tumors in murine models. Now, again, how does this pathway work? So PD-1 stands for Programmed Death 1. It was discovered in 1992 by Dr. Hanjo in Japan, and it was discovered as a molecule that was expressed in dying T cells. When T cells recognize their target, as I mentioned earlier, that's signal 1, and then normally they get a stimulatory signal to allow them to react against that target. In this case, I'm showing the molecule CD28, and this signal number 2 is a go signal. The T cells begin to proliferate. They secrete cytokines. They can kill their targets. They migrate to sites in the body where those targets are expressed. In our case, it would be sites of tumor deposits. But while this activation is occurring, those same T cells begin to express the inhibitory receptor PD-1. Now, this is a normal mechanism to turn off immune interactions at the appropriate time. So, for instance, when the immune system is encountering an infectious agent, it needs to eliminate that infection, but after the work is done, the immune system needs to turn itself off. Otherwise, you get damage of normal tissues in the vicinity. But tumors can co-opt this kind of mechanism and essentially fly below the radar of the immune system. So if the tumor expresses the major ligand for PD-1, which we term PD-L1, PD ligand 1, then if those two molecules encounter each other, the T cells will be turned off. So by blocking that interaction, either with an antibody against PD-1 or against the ligand PD-L1, theoretically we can rejuvenate the ability of these T cells, which are already tumor-specific, to kill the tumor. It's important to note that these various inhibitory pathways are not simply redundant pathways. They all have unique properties. And so in the case of PD-1, I mentioned earlier that CTLA-4 was the prototype. There are actually very distinct properties, even though these molecules are both part of the same molecular family. In immunology, I think every rule is meant to be broken. So at the risk of oversimplifying this, I would say that CTLA-4 checkpoint is more important for naive immune cells that are just learning to react to an antigen. It has a more global effect than PD-1, and you can see this by the property of its ligands, B7-1 and B7-2, which are expressed throughout the body on antigen-presenting cells, such as dendritic cells and monocytes. The PD-1 pathway is more selective for peripheral tissues, and so T cells that have been educated against tumor antigens migrate into tumors, and there then they encounter PD-L1 expressed on the tumor. Now, based on the genetic mouse models of these two molecules, we anticipated that by blocking PD-1, we might see less in the way of side effects. Because in CTLA-4 knockouts, there's such a massive proliferation of T cells that the mice die within 3 weeks of age of massive T cell lymphoproliferation. Whereas in PD-1 knockout mice, these mice live a fairly normal lifespan, and depending on the background strains, some of them develop organ-specific inflammation. But these phenotypes are very different. And in fact, what we saw in the clinic was that PD-1 blockade and PD-L1 blockade have turned out to be much less toxic than CTLA-4 blockade. So we went into the clinic in 2006 with an anti-PD-1 antibody, which is now known as nivolumab. This antibody was originally produced by a biotech company, Metarex, and this was the work of Nils Lundberg, Alan Corman, and others at Metarex. In a 39-patient first-in-human trial, we showed that this drug could have activity against tumors that were very advanced and that had not responded to other treatments. And at the same time, we saw a favorable safety profile. So that rapidly led to the trial I'm showing you on this slide of 306 patients with advanced treatment refractory cancers. Patients had one of 5 different cancer types in order to be eligible for the trial. They included lung cancer, melanoma, kidney cancer, colorectal, and prostate cancer. They had to have failed at least one other systemic cancer therapy, but in fact, 50% of the patients on this trial had had 3 or more prior systemic therapies for their cancer before they started the study drug, and so this was a heavily pretreated group of patients. We did not see any objective responses, and that's an oncology term that means either a complete tumor regression or a partial regression amounting to essentially 50% overall tumor burden reduction. We did not see responses in small groups of patients with colorectal or prostate cancer. But we did see reproducible evidence of activity in lung cancer, melanoma, and kidney cancer. And so what I'm showing you here are the response rates in those 3 different cancers. cancers, 17% for lung, 31% for melanoma, and 29% for kidney cancer. Again, this is complete or partial tumor regression. In addition, we had patients whose disease stabilized completely for 6 months or more, which is also a very good endpoint in this particular group of treatment refractory patients. And so these are additional 10%, 7, and 27% of the patients had prolonged stable disease. I'll show you some examples of the tumor regressions in response to anti-PD-1. And it should be noted that this was an outpatient therapy that we give every 2 weeks by intravenous infusion. In this particular trial, the therapy could go on continuously for up to 2 years if the patient was doing well. In this patient who was treated at Johns Hopkins, this is a patient with advanced kidney cancer. This is a CT scan, a cross-sectional area through the abdomen. You can see the spine here, abdomen here. The head would be in back of the screen, and the feet would be towards you. And so this patient had had his kidney removed, but the tumor grew back in the resection bed, and that's what you see here. It's growing through the abdominal wall and back towards the spine. This is the liver. You can see metastases in the liver as well. 6 months into treatment, now you can see a marked regression. So this is an example of a partial response. The tumor is now calcifying. This patient previously had had 4 different lines of therapy. They're listed here before he started treatment with anti-PD-1. He completed all 2 years of the therapy. He then stopped the treatment per the rules of the protocol. He's now under observation 9 months later, and he is maintaining this partial response. Now, this is something really unusual in oncology. We don't typically see patients whose tumors just sit there after we stop the drug. Usually they will grow back pretty rapidly. So again, this is the kind of finding that suggests to us that maybe we have reset the balance between the immune system and cancer, and the immune system continues to work even after we stop giving the drug. Here's another example of an advanced melanoma. This very large metastasis in a lymph node in the groin area was just one of about half a dozen tumors throughout this patient's body. This patient had a complete response to treatment, and while his tumors were going away, he developed a disorder that is called vitiligo, which is a patchy blanching of the skin, and you can see this here. Vitiligo is the result of the destruction of normal melanocytes, the precursors of melanoma cancer cells, which are responsible for skin pigmentation. And in this stain of skin biopsies from this patient, staining for melanoma, for normal melanocytes, here you see them in normal skin, and they're completely gone from the areas of vitiligo. And so this suggests that by giving this drug, anti-PD-1, we induced an immune response against this patient's melanoma that was at least partially directed against normal proteins that were contained in melanocytes responsible for pigmentation in this patient. Finally, the biggest surprise, I think, to any of us involved in these trials was the response of lung cancer. Lung cancer had previously been considered to be a non-immunogenic tumor because it had not responded to many other kinds of immunotherapy. But with anti-PD-1, we are seeing reproducible evidence of significant responses in patients with advanced disease. And this shows you one example in a patient that we treated at Hopkins. These red arrows are pointing to metastatic lesions in this patient's chest. 8 weeks into treatment, you can see that the patient already has a partial regression, and a year later we see scars where the tumors used to be. We have no idea if there may be a few live cancer cells there, and so we can't call this patient a complete responder. This patient is a partial responder. But this patient completed 2 years of treatment, and since January of 2013, under observation, has maintained this partial response. This response in lung cancer patients now opens the door to many other kinds of cancers. And I would say that we don't yet know the full profile of activity of anti-PD-1 and anti-PD-L1 drugs, but this is something that we are rapidly learning about in new clinical trials. This shows you in this trial of 306 patients, all 65 patients who had what we call an objective response, complete or partial regression. Overall, 21% of all the patients in the trial had an objective response. And what I'm showing you here is what we call in oncology a swimmer plot. There's a very long timeline here. This is the 2-year mark where everyone had to stop receiving the drug and go into observation. But you can see that some patients have been observed for 3 years or longer. The black dots show you when we saw the first evidence of a response in each of these patients. And so about 50% of them occurred at the first evaluation point, which was at 8 weeks into treatment. But you can see that many of them occurred much later. And this is a part of the challenge in using these drugs. Here we're treating the immune system. We're not treating the cancer directly. So it's an indirect mechanism of action, and sometimes it takes some time to take hold. But as long as these patients are doing well clinically, the protocol allowed us to continue to treat them. And some patients were even treated through a period of initial tumor progression. We could do that on this trial because they had not responded to other standard therapies, and many of them had run out of other options. Now, I mentioned that some of these tumor regressions have been followed for a long time, and what we find is that the majority of them persist even after the patients stop receiving anti-PD-1. And so we had a group of 27 responders on this trial who stopped treatment for reasons other than disease progression. So that could have included the fact that they developed a complete response, that they reached that maximum dosing period of 2 years, or that they developed a toxicity which prevented them from getting more anti-PD-1. But then those patients were followed. And what we found was that about 3/4 of them maintained their responses off drug for at least 4 months, but some for over a year. And so these red bars are showing you the period of observation after the drug is stopped, and the black arrows show you patients who were still in response status at the time that this analysis was done last year. Now, you would expect that with these tumor regressions, with stable disease, with prolonged responses off drug, that we may actually see an impact on overall survival. Overall survival is the gold standard endpoint for cancer drug trials. And it's a very clean endpoint because either the patients are alive or they are not alive. In order to look for survival benefit, we really need a randomized trial, and those are called phase 3 trials. That's the last phase of testing before a drug is approved by the Food and Drug Administration. The trial that I'm describing here was an early phase trial, and it was not randomized against a standard therapy. Nevertheless, we can look at the survival of these patients and compare this to literature experience and see how we're doing. And so here are the survival curves of the patients with lung cancer, melanoma, and kidney cancer who were treated with anti-PD-1, and the 1-year and 2-year survival rates. For instance, for melanoma, this overall survival duration of almost 17 months compares very favorably to the trial I showed you earlier with anti-CTLA-4, where the overall survival was 10 months. Similarly, for the lung cancer and kidney cancer curves, these curves look very promising, but now they are being examined in the ACID test, which is a phase 3 randomized trial. Now, in addition to nivolumab, there are several other pharmaceutical companies that have made drugs that block either PD-1 or PD-L1. And I'm showing you summary data here of the activity in melanoma, lung cancer, and kidney cancer. So in addition to nivolumab, there's a drug from Merck called MK-3475. And I'm showing you 2 drugs against the ligand PD-L1, one from Bristol Myers and the other from Genentech Roche. What you see is that all 4 of these drugs have significant clinical activity in patients with advanced cancer. And so this body of information now validates targeting the PD-1, PD-L1 pathway as a target for cancer therapy. But what about the patients who don't respond? So I mentioned earlier that the PD-1 pathway acts locally in the tumor. And so if we believe that the pathway acts at the tumor site, then we should look in the tumor site for clues as to how to improve this therapy. And there are two avenues of research that are very active right now that I'll address in the remaining minutes. One is to identify biomarkers which can help us select those patients who would be most likely to respond to this treatment. The other one is to identify immunologic resistance pathways and develop synergistic combination treatment regimens to overcome this. So the tumor microenvironment is complex. There are many interacting elements. You heard about this earlier this morning. On the one hand, there are tumor cells that have genetic heterogeneity and differential protein expression. On the other hand, there are very complex immune infiltrates with a variety of different T cell subsets, B cells, natural killer cells, and regulatory cells. We thought that the best factor to look at first for predicting response to PD-1 pathway blockade would be expression of the ligand, PD-L1, on tumor cells in pretreatment tumor biopsies. The rationale there was that if the tumor did not express PD-L1, then the pathway would probably be irrelevant in that particular patient. And so in the trial of nivolumab that I described earlier, we had access to tumors from 49 patients, and we stained them. This is immunohistochemistry now, looking at biopsy slides in melanoma, kidney, and lung cancer, and the red arrows are pointing to tumor cells that have a ringed cell surface expression of the PD-L1 protein. We found that that pattern of expression correlated with the likelihood of response to PD-1 inhibition. And so in our group of 49 patients with the 5 different cancers that I mentioned, if the pretreatment tumor did not express PD-L1, we saw no responders in that group of patients. Whereas if the tumor did express PD-L1, we saw double the response rate that we saw in the general population. We now had approximately a 40% response rate. This finding has subsequently been followed up by several major pharmaceutical companies which are also developing drugs in this space. They've each developed their own antibody for this kind of testing and automated methods. Each of the companies uses a slightly different definition for what PD-L1 positive is, and so it's remarkable that all of these studies are actually coming to the same conclusion, which is that if the pretreatment tumor expresses PD-L1, those patients are more likely to respond to PD-1 pathway blockade. Now, in the more recent studies, which with much larger numbers of patients, there is not a perfect correlation. And so there could be many reasons for that. It's possible that the newer tests are not as sensitive as this manual test that we were performing in our laboratories at Hopkins, but there are alternative explanations. This does raise a red flag, though, for development of this PD-L1 test as a true biomarker to permit patients to get the drug, because there are so-called marker-negative patients who nevertheless can respond to these drugs, and we would not want to deny them the potential benefit. As we were doing these biomarker studies, we learned something about the basic biology of this pathway. Our research pathologist, Janice Taub and Bob Anders, saw, for instance, in large numbers of melanomas, that whenever there was PD-L1 expression on tumor cells, in the same vicinity, there were sheets of infiltrating lymphocytes. This suggested that the tumor was putting up a defense, putting up this wall of PD-L1 expression to turn off lymphocytes that we're trying to attack. One factor that T cells secrete that we knew from laboratory studies can enhance or induce PD-L1 expression on tumor cell lines is interferon gamma. And sure enough, when we examined these infiltrates on a molecular level, we found overexpression of message for interferon gamma in tumors that expressed PD-L1 compared to the tumors that were negative for PD-L1. And so this led to the adaptive immune resistance hypothesis for PD-L1 expression, which is operative in melanomas and potentially other tumors, which is that T cells recognizing tumors secrete interferon gamma and other factors, promoting PD-L1 expression on the tumor cells, which then creates a negative feedback loop and will turn off the T cells unless we block that interaction with anti-PD-1 or anti-PD-L1. We can learn a lot more about these immune infiltrates through whole genome microarrays and directed qRT-PCR studies, and so we can actually, under the laser microscope, dissect out in a very precise way these immune infiltrates that are budding these areas of PD-L1 tumor expression to learn more about them. And here's an example of a multiplex qRT-PCR test showing that in the same neighborhood where we find PD-L1 expression, we also find PD-1 on T cells. We find markers of the infiltrating CD8 positive T cells, and we see molecules like perforin, which shows that they have killing capacity. But importantly, we also see other checkpoint molecules in the same neighborhood. And what this means is that if we block the interaction of PD-L1 with PD-1, that's fine. We'll block that pathway. But then there are other immunosuppressive pathways such as interleukin-10 and LAG3, which is another one of these T-cell inhibitory receptors, which can take over and may be the cause of tumor resistance. And so now in the clinic, we're moving to combination therapies. For instance, the combination of an antibody blocking LAG3 with anti-PD-1, it has now already entered the clinic. And this is just to give you an idea of where we're headed. The future of this therapy, we believe, is going to be combination therapies, not only with other immunotherapies, but with kinase inhibitors, with adoptive T-cell transfer, with cancer vaccines, with epigenetic therapy. Animal models in the laboratory show that these can be very powerful combinations, and now these are rapidly moving forward into clinical testing. So the field of immunotherapy has had many ups and downs over the past few decades. We would like to think that we always had good ideas, but it took a while for the technology to catch up with those ideas. I hope that the work that I've shown you today convinces you that immunotherapy has now earned its place as a fourth pillar of cancer therapy alongside surgery, radiation, and chemical therapies. And I'd like to acknowledge the people who did this work. This is the team at Hopkins, including physicians and clinicians. Especially want to acknowledge Drew Pardall, without whom we could not have done this work. And we've had many collaborators around the globe and sponsorship from many sources. Thanks for your attention.